Welcome to the Department of Genetics of the Uniwaze Academy. In this video, we will have an overview of gene therapy, and we want to answer some of your questions in this field. So, maybe you want to know about different major treatment strategies for genetic disorders. What is gene therapy? The history of gene therapy, field of gene therapy, different general types of gene therapy strategy, or something like this. In genetics, generally all topics are related to genes and DNA. As you know, if DNA gets damaged, it leads to disease. Therefore, these types of disorders are called genetic diseases. Now we want to talk about different major treatment strategies for genetic disorders. Pathogenesis is divided into two categories. The first category is related to genetic deficiency. It causes loss of function. The second category is related to disorders where phenotypes result in harmful gain of function. The treatment of genetic deficiency is augmentation therapies. The treatment of disorders with positively harmful effects is elimination, correction, or inhibition of genes. Another treatment strategy is related to preventive treatments. Until now, various strategies for treating genetic diseases have been explained. Now we want to know what gene therapy is. Gene therapy has been defined as the supplementation, correction, or modification of malfunctional genes by functional equivalent for therapeutic correction of the absence or reduced level of gene expression activity. After all, in order to talk about gene therapy, we need to know its history. In the picture, we can see the path of gene therapy during the last century until now. It can be divided into 4 phases. Basic research phase lasted for 60 years. In the second phase, early age of the gene therapy practice was beginning of clinical trials. In 1990, for the first time, gene therapy was officially and successfully performed on a 4-year-old girl who had an enzyme deficiency. Make sure to subscribe to Uniwaze for more captivating insights into the world of science, medicine, medical laboratory, and innovation. Despite the small successes in the second phase, an unfortunate incident also happened. In 1999, a young boy with an enzyme deficiency died due to an immune system reaction. The third phase is related to starting the approval of gene therapy products. Here you can see the list of products that were produced in the third phase. Gendesin, the first gene therapy drug to be approved in China. Since then, more than 3,000 people suffering from head and neck cancer have been treated with this medicine. The fourth phase of this path is related to the future of gene therapy. Make sure to subscribe to Uniwaze for more captivating insights into the world of science, medicine, medical laboratory, and innovation. Now we understand that gene therapy is as old as a century, so let's talk about fields of gene therapy. As you can see in the diagram, gene therapy has been done more in field of cancer than in genetics or infectious diseases. In the field of cancer, hematological cancer has a greater contribution. And in the field of genetic disease, metabolic disease has a greater contribution. Now we want to know about gene therapy strategies. Gene therapy strategies are divided into 2 categories. The first category is related to strategies that modify the cell. For example, gene augmentation, gene silencing, repair of mutant gene. The second category is related to strategies that kill the cell directly or indirectly. Gene therapy strategies require vectors for gene transfer. Ideal gene therapy vectors should have high transduction efficiency, the ability to induce long-term and stable expression, enough capacity to transport large transgenes, be specific for target cells, be able to transduce both quiescent and proliferating cells, and avoid random insertion and mutagenesis. Different vectors with different characteristics have been used for various applications. The most common type of vectors is viral vectors, which can effectively transfer their gene into their host cells. Three important viruses in gene therapy include lentivirus, adenovirus, adeno-associated virus. The advantages of viral vectors, including higher transduction efficiency, greater ability to be engineered, and highly specific gene delivery, have resulted in a wider range of applications when compared to non-viral vectors. In this video, we have an overview of gene therapy and learned about its history and strategies. Thank you for watching. Please don't forget to subscribe to the Uniwaze channel.